Hi, my name is Chen Kam, and I'm an assistant professor here at UCLA. My lab studies the vasculature, which is the vast network of blood vessels that makes up our circulatory system, allowing for the transport of oxygen and nutrients via the bloodstream to all of the organs and tissues throughout our bodies. We are really interested in understanding the regulation of the vasculature within the context of the native tissue environment, or in other words, how the vasculature functions within a living, breathing organism. In order to do this, we apply a specialized microscopy technique called multi-photon microscopy. And this microscopy technique allows us to see directly into the skin of live mice in a completely non-invasive manner. And when we couple this with genetically modified animals that express fluorescent proteins in their cells, this allows us to watch the behaviors of the cells that make up our blood vessels within a variety of contexts, whether that be in the process of development, in specific disease models, during the process of vascular regeneration, or even aging. One area that we are really excited about is understanding the process of vascular regeneration. Given the crucial function of the vasculature, blood vessels have evolved rapid and efficient mechanisms of regenerating themselves following an injury. However, there's actually still a lot that we don't understand about this process, such as how, when, or where specific populations of vascular cells are able to coordinate their behaviors in order to actually regenerate new blood vessels following injury. We hope to apply our imaging technique to gain a deeper understanding of the fundamental principles that allow for the process of vascular regeneration to occur. And with the hope being that we're able to leverage these insights to be able to improve the process of vascular repair and regeneration in conditions where this process is impaired. One example of this is in chronic wounds, or wounds that have trouble healing, a very prevalent condition that is often associated with diabetic ulcers. And we hope that our insights will be able to allow us to develop therapeutics to hopefully improve this process for afflicted patients.